Hello there everyone. I am Sigrid.
Good day to you guys. Well, I am Jessamine. And welcome.
And today we are going to present to you how routine urinalysis is done in the laboratory.
You guys excited? Well, come on, let's go. And watch how it is performed.
Let's go inside the laboratory now. So, before going to the work area, there are standard precautions we need to follow.
Like what it is shown right here. These are called personal protective equipment, which includes hairnet, eye goggles, laboratory gown, disposable gloves, and surgical face mask, which minimizes the exposure from hazards. Another important thing to remember is to do hand washing properly.
Hand washing is a very crucial step to sanitize hands and must do after touching potentially infectious fluids and items, or before and after touching patient's environment, or contact with patients.
So, we are now in the laboratory. We are Sigrid and Jessamine again, and we are here, in our working area. The very first thing we do is to assemble the materials and equipment.
These are the materials and equipment needed for the test: plastic conical tubes, test tube rack, patient's sample in test tube, glass slide, amp, cover slip, refractometer, distilled water for calibration, microscope, micropipetter, with disposable tips, paper tissues, surface disinfectant, reagent strip, worksheet, reagent color chart, and centrifuge.
There are 3 examinations involved in the routine urinalysis. These are physical, chemical and macroscopic examination of urine.
So, let's do the physical examination. Let's go, guys. After labeling the patient's sample, observe the color of the urine and the transparency, then record it on the worksheet.
In a closer look, this is a urine sample, and it has deep yellow color, and its transparency is clear. And now, we are going to perform reagent strip test for the chemical examination. We need here is the reagent strip with its color chart. Then dip the reagent strip into the test tube with uncentrifuged urine, moistening all pads. Remove the strip immediately, then blot gently the edge of strip on the tissue. Then start the timer, as it is withdrawn from urine.
This is the crucial step in the reagent strip test we are doing a while ago. It's blotting the reagent strip to avoid or prevent run-over. Observe the reagent strip and compare the colors to the color chart at appropriate time intervals.
And record the results on worksheet. Then we can proceed now in the microscopic examination. Let's go, guys. This is the last step for the routine urinalysis. Before proceeding to microscopic examination, we first need to centrifuge the urine. Transfer the urine to the conical tube using micropipetter, for about 10 ml of urine volume.
Centrifuge the urine and balanced it with water on another test tube, and set it for 5 minutes at 1500 rpm. After that, remove the conical tube and pour off supernatant urine. Leave only approximately 0.5 to 1.0 ml of urine in the tube.
We are now finally on the last step of routine urinalysis. So, after resuspending the urine sediments, we are going to place a drop of urine in glass slide, and putting the cover slip on top of that.
Place the slide on the microscope stage, then focus it on low power field. Adjust the light and condenser.
These are the sediments examined under the low power objective. Epithelial cells and up to amorphous sediments are reported semi-quantitatively, like describing it in few, moderate, abundant, occasional, or many, while cast sediments are reported in range, per low power field.
And we are going to show you examples of urine sediments in the microscopic slides. And first one are ones found in low power field. This part is the squamous epithelial cell. And the other slide is the transitional epithelial cell.
This slide shows crystal sediments. Then on the other hand, is the example of thin lines of mucus threads.
And this is the microscopic look of waxy cast.
And, next is for the sediments seen in high power field. This one is the white blood cells.
From the top, the slide is the renal tubular epithelial cells, and from down right are the yeast cells. And lastly, from its left is the bacterial sediments. Then record all the results of the patient's sample in the worksheet provided.
After everything is done, make sure to clean the work area, and discard all the used materials in their designated hazardous trash bins. Clean also the equipment and return it properly to its storage.
Removed also your protective equipment, and throw it properly in yellow trash bin. Lastly, wash your hands.
Then that is all, guys. Thank you for joining us in the demonstration of how routine urinalysis is performed. We are Sigrid, Terry and Jessamine Birake. See you then.
Hope you enjoy watching how routine urinalysis is performed. Goodbye.